So firstly, stripe rust. I'll take you through pathotypes, industry issues in 2010, and some sprayed versus unsprayed NVT trial work. This has got very complicated over the last couple of years. So certainly we had stripe rust around late 70s, early 80s. You can still find that pathotype at low levels out there, but basically we moved to resistant varieties and that didn't become an issue. What happened in 2002, we got the introduction of an exotic stripe rust pathotype. They believe it's out of North America. We've run into problems, and that's still representing 13% of what's around. Particularly caused a problem in varieties such as H45, Baxter Lang, and a range of others. Unfortunately, when you've got big numbers of spores there, you get mutations, you can lose other genes. I guess the one of most significance, we lost a particular gene called YR17. Stripe rust is called yellow rust everywhere else in the world. Took out this YR17 gene, and that's certainly dominating what we saw last year. That caused a problem for varieties Ventura, Crusader, and a range of others. Unfortunately, again, I guess we've had a stepwise mutation. So we've gone from here, taken out one gene, this one again has mutated, we've lost a second gene called YR27. Okay, so it's combined virulence for both. That happens to be in Livingston, Morinda, and Ruby. So they're varieties we need to keep an eye on. Certainly they have dropped down in their resistance. How far they drop, we don't really know. We had a separate mutation from here to this Jackie, really a problem more for the triticales, Jackie, Breakwell, Kosciuszko, and further mutations through there. This Tobruk one, which is round 09, saw a lot more of it, 7% last year, has taken out a further gene that's mainly in the triticales. The key thing, but I guess coming back here, where you've got the YR17, YR27 pathotype, it's going to infect all these varieties right up through here. It's got virulence for all of those, so that's a particular issue. There is another pathotype around this, actually an old one, which takes out YR4, which is in Lincoln and Bolac. And the only good news, I guess, is barleygrass stripe rust, which we see, it doesn't actually infect wheat. Industry issues in 2010, okay? This is a photo that was sent to me by email from a very good agronomist around Canamble, won't mention his name, sent me an email, hopefully you can see it, plenty of yellow through here. My Gregory's fallen over with rust, it's getting smashed. Okay, I sent him, had a look at the photos, sent him back a reply, said, look, the flag leaf's a bit wide for Gregory, and you could actually zoom in, he took a high enough quality photo, you could see the auricles, which is the collar around where the leaf joins, and they weren't purple. Gregory's got a purple auricle, okay? So I questioned him, I said, are you sure this is Gregory? And got a pretty quick reply back saying that he swore, yeah, on email, saying, you know, well, I've got 15,000 bloody hectares of it, of course it's bloody Gregory. 3 days later, I got another sheepish email back saying, actually, you're right, it was Baxter. Okay, this was just a mix-up. But the problem, the issue I've got with this is that then goes around the pub circuit, and this went round the Canamble district in that 3 days until it got corrected widely. Everyone had lost confidence in Gregory when it was just a straight variety mix-up. And the problem for the grower, he thought it was Gregory, he managed it like Gregory, and ended up looking like this when it's Baxter. And the agronomist did the right thing too. He, in all good faith, he was told it was Gregory, so this is what happens, and that causes issues. This was not the only case last year, it was widespread, especially with Sunvale and EGA Gregory S varieties. As I said, Gregory's got these obvious purple auricles around the collar here. There's a bit of a giveaway. There is a low-level impurity in Gregory. It is not a contaminant, it is just not homozygous around a particular gene. So it's got a bit of variation. That is well under 5%. And those plants that are the off-type will take it worse than what the true Gregory does. Fortunately, the off-type doesn't have a purple auricle. It has a white or clear auricle. So if you've got a Gregory crop, you think it's getting smashed, have a look at the colour of that auricle. If it's the off-type, then that's the issue there. Sunvale is another one. People probably got mixed opinions on this. Sunvale has a very characteristic head type. Very compact, small heads, not much gap between the spikelets as you run up the head. Okay. There was a lot of Sunvale that I saw last year that took a lot of rust, but it wasn't Sunvale. The head type was just not right. The classic was up north of Moree. We got called out into a crop, shown up at the update there at Narrabri, I can't remember what it was, at Moree, sorry, a big issue. When you looked at it, there was a pattern. There was a lap around the edge of the paddock, and there was a couple of machinery runs up the end. And they were the ones taking the rust, and they didn't have the head type for Sunvale. The Sunvale that was inside that was clean as a whistle. So when we challenged the grower, he finally looked his records up, he changed varieties at 3 in the morning. And that's what I said to the grower in the paddock. You've got to go back to the pub, tell all those people you've told that sunbale's now a sucker for rust, you've got to set them straight. And this is the problem. These things get through the industry, gather a life of their own, and we always lose confidence in resistance, which is a big issue. I'm happy to stand here and say I looked at a number of MVT trials that are sprayed and unsprayed. So in the unsprayed replicates, where you've got true sunbale in NVTs, true Gregory in NVTs, they took very little rust, and Richard's data certainly there, last talk, highlights that too. And there was certainly plenty of pressure in the trials. The susceptibles in those trials were yellow from top to bottom, so they are a good resistant source. I guess this is the tip of the iceberg, where a grower's going to get their seed in 2011. Stripe rust head infections were certainly widespread last year as well. You can get it in behind the glooms here. Basically, you only get the infection while the crop is flowering. So it infects through the florets. Once it's finished flowering, you don't get any further head infection. Tend to get it in more susceptible varieties. It's all about pustules in the upper canopy. If you've got pustules in the upper canopy, you can get a head infection. Even the most resistant variety can still get head infection, but that won't happen usually commercially. Head sprays, very different target, and certainly by the time you see it, they're ineffective. And you've really got to wonder, once you get to this stage, you're going to get your withholding anyway. Moving on to the sprayed versus unsprayed NVT trials. So this was an initiative that was taken through the National Variety Trials funded by GRDC. I&I New South Wales happens to have the contract for New South Wales. There was 9 sites where they had extra reps. They sprayed 2 reps and they left 2 reps unsprayed. And this is more to do what growers and advisors, I guess, are reluctant to do commercially. You see a bit of rust in the crop, the temptation's there to spray it. With these, they let them run and let it get as bad as they can, so it's actually good data for that. There was 9 sites throughout New South Wales. I've deliberately done an average across those to take out some different effects. Okay, a few ground rules. Last year was a very conducive year. We had big yields. We had the option to convert any leaf area loss into yield differences. Stripe rust in the 4 I saw in my region, Dubbo above, certainly were driven by stripe rust. I didn't see the ones down south. There may have been a minor yellow spot component to it, but I don't think so when you look at the reaction of some of those varieties to yellow spot. And they all seem to be driven by the YS17 pathotype. The other thing to emphasise, with the spray program you've got, you have some green leaf retention activity with these sprays. We usually don't convert green leaf retention in the north into yield gains because we don't have the soil moisture to convert that. But last year was certainly a situation we might have. So if you love spraying, this is as good as it gets, okay? So this will probably be on every chemical company brochure shortly. So how bad did it get? This is Trangie, MS variety at Trangie. Mightn't come up so well, but yeah, certainly very yellow, so a lot of rust pressure. I only looked at the main season. Well, Alan actually did it, but we looked at the main season spray timings. The early timings on the early entries was just wrong, but the control in the main seasons was very good. We broke them up into resistance classes for the YS17 pathotype. We've only had one very susceptible, Col 2000, and you can read what we've put in our analysis here for MRs, etc. Cutting to the chase. Blue is where they were sprayed, red's unsprayed. So in a very susceptible variety, we lost 64% yield. 29% in MS to S, 31 in MS, 26 in an MR to MS. We take one variety out of that, which you'll probably see, and that drops to 19. 11% in MR, so it certainly drops off as we go up resistance, which is to be expected. Looking within the moderately susceptible varieties, certainly where we control it, we can get yields up that 4-5 tonne plus. We have differences in that drop-off. A bit of a no-brainer. Moderately susceptible varieties, you've got a big difference between your red and blue. It's worth spraying. MR to MS, certainly a big range again. You probably, I won't point the thing in it. One particular variety probably performed worse than the others and certainly had a 2 tonne difference there, but certainly a range there. But certainly those, with the exception of this one, the bars are getting closer. You run into the MRs, certainly some yield losses without spraying or yield gains from spraying if you want to look the other way. Not in bounty, questionable, but certainly ranges there. Economics, so what we've got up the side here is the yield gain from the spray program, okay? So we actually picked up over 3 tonnes in a very susceptible variety by spraying it compared to unsprayed. Our economic assumptions, $200 grain price and a fungicide and application cost of $51, okay? All these trials had 2 sprays, generally a full rate of tebuconazole at 32 and a full rate of epoxyconazole at 39. You cannot guarantee these yield outcomes unless you do exactly this. Okay, and it gets more expensive with some of those by plane. So you can't assume you're going to get this yield benefit from spraying with a low label rate one-off spray. Everyone happy to accept that? That's your break even, so if you just want to fill your time in, you would have done that with all of them, and that's your $2 return on $1 spent. So you're not getting it here in the RMRs, and you just managed to get it in the MR varieties in some of them. So remember, this is as good as it gets. The question's always been, is it economical to spray MR varieties? Certainly out of last year, some of them it was, but you're going to need a season like 2010 to do it. Okay, it's still going to cost you the same amount for that program in a dry year, but you're not going to get your half-tonne benefit on a 2.5 tonne crop. And I think the debate still is, we've stood up here and said it a million times, you do not increase yield when spraying for stripe rust. Okay, you're protecting yield potential, and it comes at a cost. And I guess where the debate doesn't go, that $50 or $30 or whatever you spent, what have you spent that someone else in your program, maybe on nitrogen top-up last year, in a resistant variety, would you have been in front? Oops. Quickly, I'll go through some, if I've got time, some head infection work out of last year. I won't cover the first bit on what they actually look like. Stephen Need will do that last talk. Bit of work we did on distribution of species, and I guess just to highlight some issues that I'm a bit concerned about for sowing 2011. So I got 108 samples into Tamworth last year. We're actually talking two different diseases. We've got this Botryosphaeria head blight, and we've got Fusarium head blight. So the 108 samples we got in, 65% of them were Fusarium head blight only, in the blue. 6% were Botryosphaeria only. 22% had a mixed infection of both Fusarium head blight and Botryosphaeria, and 7% were other, they weren't a head infection. Inevitably that was either frost or stripe rust head infection. Where the samples come from, 77 come out of northern New South Wales, 30 out of southern Queensland, and I got one from Victoria. We actually got Botryosphaeria out of 30 of those. It might have been a mixed infection with Fusarium head blight, but as you can see, 22 of the 30 come out of southern Queensland, only 7 come out of northern New South Wales, and the 1 Victorian. So when you look at that, we only got 30 samples out of southern Queensland anyway, 22 were Botryosphaeria. Actually, 73% of the paddocks we looked at had Botryosphaeria infection, whereas only 9% in northern New South Wales. So certainly, as consistent with other work that's been done, it was mainly a Queensland issue. We're also interested in which actual Fusarium species is causing the problem when it's Fusarium head blight, okay? You can have two species. Fusarium pseudograminearum, the FP here, which is the crown rot fungus. So you can get infection at the base of the plants, under continuing wet weather you get spores produced around the base of the plants that can splash up at the head, cause a head infection, or the crop lays over and comes in contact. The other one in red is Fusarium graminearum, which is, well, more the true head blight fungus, has a perithecal stage on corn and all the winter crops and grass, etc., and produces an airborne spore that lodges up in the heads. So very different for northern New South Wales. When we look at our head blight samples, 72% of them were Fusarium pseudograminearum, so the crown rot fungus being the main issue. Only 9% were Fusarium graminearum alone, and 19% had a mixed infection with both species. Southern Queensland, very different. Where we had Fusarium head blight there, 80% of it was graminearum only. Only 8% was pseudo only, so crown rot caused an issue, and 12% was mixed, so quite a different picture, and that's consistent with other work too. I guess for us in northern New South Wales, I'm certainly concerned of that for crown rot this year. I think what happened last year was a real warning for us on what levels of crown rot was sitting in crops. I went back through my diagnostic book in June. I got a sample, Birchie won't mind me saying, Brenham wheat, that in June got sprayed out because it had that bad crown rot in the base of the plant. And also got sent in a sample of oats that had very severe basal browning in June, just after 3 weeks of dry weather. So I think we're sitting on a big crown rot year last year. We ended up with a really wet year, which we don't get the expression of the whiteheads, and we got a bit of head blight instead. So certainly a concern. The other issue is seed quality with some of this grain. So certainly Fusarium infected grain in particular causes a seedling blight. If you sow it, you get a really severe crown rot infection and can kill the plant as it comes out. This is just some grain samples I've got in recently. So visually when you look at it, 1% pink grain down to 18% pink grain, but when you actually plate from it, you can cover a lot more of the seed has actually got Fusarium inside it. So if you get later infections with Fusarium head blight, The grain looks normal, but the fungus is still in it. If you sow it untreated, you can take it out with the seedling disease. We look at our germinations here, this visually better stuff, better germination. You get down here, it looks horrible, the plate's out horrible, and it's going to come up horrible. So you probably want to look up some of that sowing density work. What's it actually look like? 1% Fusarium, 99% germination, 8% Fusarium, 74% germination, vigor's still pretty good. 39% Fusarium isolated from the grain, we're down to 40% germ. You can start to see a lot of these getting colonised, and this is just horrid. And certainly germination, when you see Kev Moore's work on the chickpeas, germination does not necessarily equal emergence. I don't know how well that comes back where you are, but certainly if you've got grain that looks like this, just don't sow it in 2011. The other one is seed quality with Botryosphaeria. So this is a seed lot from Swan Hill, 50% white grains. You can see the white grains in here. So what I actually did is just pulled out 100 from that same sample, 100 brown grains, 100 white grains, and just did a germ test on them. That's what she looked like after 3 days. So this is your Botryosphaeria, very rapid grower coming out. That's what she looks like at 10 days. So we've still got 48% germ. A lot of these are very slow to germinate. Some of them weren't too bad. I don't know how this is going to go when you put it in the ground. Last slide, crown rot, okay? We've got a heap of samples to process over at Tamworth. We've done a few of them so far, and this is what a fair few of them are looking like. They've got very severe basal browning. I just wanted to make that point. So crown rot infection loves a wet year. It needs moisture to infect. So we've had plenty of infection events last year, a lot of basal browning, a lot of colonisations. The expression in terms of whiteheads hates a wet year. We need that moisture stress to get those whiteheads expressed. So just because you didn't see whiteheads doesn't mean you haven't got a crown rot problem. Certainly big yields last year means bigger stubble loads, so you need less of it to actually have the crown rot fungus in it, and you're back to the same point. And what are we going to do in 2011? Okay, we know breakdown of stubble is critical to displacing the crown rot fungus. Certainly some of these early sown chickpea crops that produce big biomass, run into botrytis grey mould problems, should have decomposed a lot of that stubble, and probably our safer options, lower levels to go back into. Stubble management will be an issue, what we do with that. Dare we mention burning may be an option. It'll only get rid of your above ground portion. If you had high levels in the crowns, it'll still survive in the crowns as well. And certainly if you've had floodwaters, you've moved stubble and it's got the crown rot fungus in, you've moved the inoculum source. Basically, I predict dry spring, I think we're going to have a disaster with crown rot. So I'd be using your stored water wisely. Probably not a year to chase your highest yields. Be a little bit conservative, conserve a bit of water for grain fill. That's me.
Steve, how do we conserve moisture for grain?
I think this is where we target our yield, I guess, not putting all our water into early biomass. So be a bit conservative on any nitrogen and maybe your sowing rates, etc.